All right, folks, welcome back. Just by way of announcement, next week is our first test. That'll be on chapters 1 through 4. And before the test, there is a quiz. The quiz is available on Canvas. So if you sign into Canvas on Friday, Saturday, Sunday, or Monday, there'll be a quiz. Quiz 1. It's 10 multiple choice questions. It's timed, I think, for 15 minutes. Open it up, proceed through, click on the answer, submit it. Low number of points, that's 1% of your grade. So don't view this as like a high pressure type of thing. All right, do some studying, watch the videos. Read those chapters in the textbook if you have it. If you don't have the textbook, you can go right to the library, go down to the bottom floor. It's on reserve over there. And maybe leaf through. Particularly, I would say chapters 1 and 4 are going to be the ones that have a bulk of the stuff that I think is important. And take the quiz and see how you do, right? The test next week will be in person, so just come in as usual on Thursday and we'll have the test. So questions about that? Okay, I want to start out with talking about the Lizard Lab. And oops. Okay. I hope everyone was able to watch the video from Tuesday. If you don't watch those videos from Tuesday, the Thursday lecture may not make a whole lot of sense. So a lot of times we're building ideas from Tuesday's lecture. This, I know you guys have gone through and done this, but I just want to kind of reflect back on this. This fits really well. The modules 3 and 4 from the Lizard Lab really fit well with our discussion of basically Tuesday we talked about evolution. Right? And you can think of this as kind of microevolution. Changes at a small scale, changes in the genetic makeup of population on sort of small things, right? And then today we're going to talk about the speciation, right? And that is really talking about the cumulative effects of lots of small changes in the genetic makeup to the point where two groups split enough to be different species. Okay, so this is kind of like macroevolution. Okay. Module 3 was really about microevolution, right? Talking about these islands, the Bahamas. Always surprised how far north they are, right? For me, there's even further north than that. Here's Florida, a bunch of islands. It's a really good place for experiments. Talking about this Gran Abaco Island. Off the coast is this little iron K. There are naturally occurring Anolis crystallitatus, I think it is, live on the island. Okay. No, actually, Sarsaparilla. This is the brown anole. So what scientists did, they took a breeding pair of anoles from iron K, and they moved them to these 7 islands. The good the good setup here, or the reason that this is a perfect place for an experiment, is they knew that there were no pre-existing anoles on the island. So they bring them out there, they let them do their thing, and then they go back we'll see and measure aspects of those new populations. Iron Cay is a bigger island. It has multiple habitats. It has bush, grass, ground. It has trees, right? So there's going to be the canopy, the trunk. These little islands don't have the same habitat. So very different environment, okay? Out in this environment, it's basically just small bushes and grasses, okay? Key point of this, a couple key points. One, there were no anoles here. The anoles were transplanted, okay? Allow a couple of generations. So for 4 years, they go to these islands and measure the leg length on these new islands. So obviously, who are the lizards that they're measuring here? They are the great-great-grandchildren of the original founders of that population. Okay, so we know genetically where they came from, and we're transplanting into a different environment. Okay. So, this is kind of a refresher for you here. That's module 3. So if we look at the answers to these questions here, right? After several generations, they measured the leg lengths of 10 lizards, which this is not really that important. Which group of lizards have longer legs on average? Right, and I don't really expect you guys to remember the specifics, but clearly you can see here what happened to the leg length as the populations colonized these new experimental islands. Decreased, right? Now, a couple this is a little bit more subtle question about that data. I think you guys can all clearly see the average was lower on that graph of the experimental islands. But what can we, how else can we get a little bit more specific? Every lizard on the experimental island has relatively shorter legs than every lizard on Iron Cay. Let's go back to that graph and tell me how you can see on the graph that A is not true. Here's the experimental island, here's Iron Cay. How can we see on here that not every single lizard on this island had shorter legs than every lizard on this island? What do you see on here? That tells us that just ain't true. Okay, well, let me ask you this. What does this point represent right here? The top of this graph. And this number here. 0.829 and 0.808.
Is all of them added together and is the mean of...
That's the average, right? So probably there was no single lizard that had exactly this relative leg length, right? Okay, perfect. What about this and this? What do these points represent? What does this bar represent? What's the way to conceptualize it? If you see a graph like this and you see those bars, what information do you get from these bars? I mean, the technical answer that really doesn't tell me anything is that they're error bars. But what does it represent? What does it mean? So this is just some fictitious average. What about these error bars? What do they tell us about the data?
That it's between them, that amount, 0.84.
Yeah. This is not exactly to say that this was the smallest leg length, right? And that this was the biggest. But this is like 95% of lizards fell between here and there, okay? So in other words, there were some, even on Iron Cay, that had pretty short legs, right? And if we look on the experimental island, some of them had pretty long legs, okay? So between here and here, what would that be? That would be overlap, okay? To say that some have short, some have long, some people are tall, some people are have really curly hair. Some people have anything that's genetically determined. We see variation. And generally, what does that look like? It looks like this. All right. We see this nice bell-shaped curve. So that's kind of what we're seeing here is, here's the bell-shaped curve for one island, like this, and here's the bell-shaped curve for this island, okay? Here's the exceptionally short-legged, well, I don't know how you could actually have cereal here, but here's the exceptionally short-legged ones on Iron K, really long-legged ones, so on and so forth. Okay, that's just biology. Any trait that's controlled by multiple genes follows this kind of trait. So we know that, yeah, we can clearly say that A is not true. What about B? What would you say about that? The answer B. Lizards on experimental island have become a different species from lizards on Iron K.
So that's not what we're talking about. Different or not.
So, where did that come from? You can't see that from the graph, okay? I don't see anything on there about different species. That's something totally different than that. Now, could it be that they have become a different species? I don't know, maybe, but that's not what we're talking about here. What about C? There is no variation in leg length among the lizards on the experimental island. There is no variation. Clearly, there's actually a lot more variation on the experimental island than there is on the Iron K island, okay? So that process of elimination gets us to D. On average, the lizards on the experimental island seem to have shorter legs. See, there's even some weasel words in here. We're not real confident about this, but it appears that they have shorter legs, okay? You guys should get comfortable with looking at data like this. Because, again, you're probably not going to be an animal person, but if you go into, pharmacy, and when you're talking about the efficacy of a drug, then it's going to be the same stuff, same type of figures that you're going to be looking at to make decisions based on. This species of lizard used in an experiment is a trunk ground anole. Okay, and what do we know about those? Trunk ground. Remember we had the grass bush, the canopy, the trunk ground, and the twig. What do we remember about these guys? What was their niche? They lived on the trunk at the bottom near the ground. How do they make a living? They hang out on the trunk. They're ready to jump off and snatch up their food off the ground, right? So they would be this guy there. They perch on the broad tree trunks and run fast on the ground. So they start out as a long-legged trunk ground species. That's important. When they get moved to the island, they had shorter legs. Why? Right? How do you explain this transition from here to there? Why is it likely that we saw evolution? What about A? Is there any evidence that the ones on the Iron K just were unusually long-legged? No. B, again, that's the one you can always, every multiple choice test you take, look for answers like B that are just totally out of left field, cross them off, right? C, selection for shorter leg on the experimental island and for longer legs on Iron K. What do you think about this one? The difference here is we're saying there's selection here, there's no selection there. That's what we talked about on Tuesday. What is selection? What is this idea of selection? Anybody watch that video? I can't tell you to do that, I can't make you do it, but I'm gonna really encourage you guys, before you come in on Thursday, just try to take that time to watch. Selection is basically, it is where there's an advantage, so that the organisms or the lizards we moved, and let me see what the next question is. Let me just talk about this real quick, because this is a really, really key, important point in this class. Okay, so here's Iron K, here's the big island. All right, here's the experimental island. Say these are the anoles that we moved. Well, no, actually, we just moved 2 individuals out there, right? So here's mom and dad. They move out here. And they have, say they have 20 babies, okay? And those 20 babies have this range of leg length, right? They have, here's little Johnny with just short little legs. It's just ridiculous. And here's Sally with this. This is our 14-year-old. She's all legs. It's like this much and huge long leg. Okay, now when we move them out to the experimental island, who's going to do better on this island? Remember here, we have these big trees and bushes. This is where mom and dad lived and where they made their living. When they get out here, there is no big long tree. There's just little bushes, okay? So who's going to do better out there? Long-legged Sally or short-legged Johnny?
Short-legged.
Okay. Why? Because the environment's different. And if you have long legs, it's just awkward and ungainly trying to navigate through little bushes, right? So there's selection. Little Johnny does better, and so therefore he is more able to get food. He's able to build up a lot of fat stores, right? He is able to have more offspring than his sister, okay? And what do you guess, right, about the short-legged dad? What are his offspring going to be? This or this? They're going to have short legs or long legs? Well, we know the answer, because here's your answer. These are little Johnny's great-grandchildren, and we know that they have on average shorter legs. But why is that? Because it's DNA. He passed on his genes. That trait is genetically determined, okay? And this is natural selection. If you have a trait, there's variation, right? There's short and long leg, and then there's the average people. If you have a trait that can be passed on to your offspring that gives you an advantage, then that trait will become more and more common, okay, in the population. That's selection. That's evolution. That's what we're talking about here. Okay. Yeah, the answer would be C here, that there's a selection for shorter legs on the experimental island. Why? Because in that environment, they are better able to acquire mates and pass on the genes. What does this data and evidence suggest? A, again, what's this thing? We're going to talk about that in module 4. Cross it off. B, in different environments, different variations in trait provide an advantage. Can we cross that one off yet? No, don't, that one seems good to me, but don't answer it yet. Let's read the other ones. They might be even better. Evolutionary change occurs over thousands or millions of years and cannot be measured. Why is that one wrong? Because what kind of time scale are we talking about here? What is it, 4 years, right? Cross this one off. That may be true, well, except for the cannot be measured part. What about D? Over time, lizards will evolve shorter legs regardless of where they live. Right? Well, that's just ridiculous, because, yeah. So B. Okay. And then 6. If you place a lizard from Iron Cave on one of the experimental islands, its legs will get shorter over its lifetime. This is a really, actually it seems silly. This is a really important idea. True or false?
False.
False. False? What do you mean? Here it is right there. It's right there.
That's over the 4 years with the different.
Is
Offspring.
this the same animal as this? Is this the original animal?
No.
No. These are the short-legged offspring, great-great-great-grandchildren. So evolution happens at the scale of a population, okay? People don't evolve. Animals don't evolve. Populations evolve, okay? Because we hope like hell our population, our genetics don't change over time, right? The DNA that we're born with is the DNA we die with. Our legs don't get shorter in response to the environment. Our offspring have different abilities to survive and reproduce based on the traits that we give to them, okay? Very key point. We always think, that something evolves in order to, this would be like saying, right, you arrive on the island, you look around, you're like, oh man, my long legs are really not gonna cut it out here, so I'm not like, I'm gonna just try to shrink them up, right? It's not how evolution happens. Evolution happens over generations of time, differential survival reproduction, okay? Okay. Module 4, right, we see one species has brighter dewlap. Just want to, again, show, here we have, this is the actual data from the module. Here's 2 other hypothetical data sets. Okay, what's the difference between this graph and this graph? Actually, I should have kept this at 4.5 over here too. But in which one can you say, can we say that 4.8, oh, that's why I did this, 4.8 and 4.5, are they different from each other? Can you say that this one is larger than this one? What about this one? 4.8 and 3.9. Do you see a difference between those two? What about this one? Do you see a difference between these two? How do we determine if there's a statistically significant difference between 2 numbers? There's 2 things we're going to look at. One is the average. Is the average different here? Yes. Right? Is the average different here? Yes. But the other thing we have to look at is the variation. Right? See how small the variation is here. There is very little variation in either of these populations. So I would say yes, these 2 are different because these don't overlap. There's no overlap between those 2. Over here, the average is quite different, but look at how much variation there is. This may not prove to be statistically different because of all this overlap. From here to here, there's a lot of overlap. Right? So just be looking at that variation. Why would the dewlap color of lizards change through time? Why is it an adaptive type of thing? The color of the dewlap. Some species have bright dewlaps to make them easier to see, and others evolve dark dewlaps because the dark dewlap is better camouflage. Okay, you're shaking your head no. Why is that?
It's not for really camouflage.
Okay.
For mating.
All right. If your point of this thing is to attract a mate, why would you want to make camouflage, right? That would be like, that would be like, okay, I'm gonna do my hair up real nice and then put a hat on, right? It just doesn't make any sense at all. B, species with bright dewlaps have brighter skin. What the hell? I don't know what that is. Different lizard species have evolved different dewlap colors by chance. Well, that may be true. There's nothing to do with this. So If it's a shady place, bright colors work well, which that's sort of intuitive. But the flip side of this, which is a little less obvious, is if you live in a very bright spot, then actually dark dewlaps work better, okay? Because they contrast with the really bright environment, okay? Now, we'll see why this is important. So yes, obviously, the color of the dewlap is an adaptation that it was produced by natural selection in the same process we just talked about. Right? You've got really dark ones were able to secure a mate because they had dark dewlaps in the bright environment. So it's an adaptation to the amount of light in the environment. Okay, so this another species, Anolis cristallus. It lives in the dark, shady forest. And so, was this consistent with this hypothesis? Is the results that you got consistent with a hypothesis? They live in a shady place. They have a brighter dewlap color. So is that consistent? Yeah. Yes, because it's easier to see in the shady environment. Okay, what about this one? These two anoles, are they different species? Are they different ecomorphs, different morphologies, or different genera?
Species.
Okay, how do you know?
The name is different.
Okay, because they have different names, right? This is the genus name, Anolis, so you can cross that off. They're both in the genus Anolis, and that they have different named specific level taxonomy. So yes, they may be different ecomorphs, I'm not really sure, right? But we know they're definitely different species. If you go into one of these areas in the Caribbean and you gather up all the anoles and you look at them, they will have different colored dewlaps. Why is that? Why are they partitioned out? That's part of their niche, maybe you could say. Why do they have different colors? Why is there not 3 species of anoles with orange dewlaps?
So the mate doesn't get them.
Yeah, we want to keep the species reproductively isolated from each other, right? And so it keeps the species reproductively isolated. Okay. So this does end up fitting into, it's a little bit more difficult since you guys did the Anole lab on your own. I just want to make sure that we're making the connections between what we're doing in lecture and lab. So, lots about microevolution. Today we're going to talk about large change evolution. You can see this best because it does tend to happen on longer time scales. It's really best observed in the fossil record. Right? And again, tying this back to our discussion of phylogenies, right? And we talked about these family trees at length. And what are we assuming is happening at each one of these points I'm circling here? What is this? This is where we have a divergence or a splitting. We have one group splits into 2, that group then splits into 2, and so on and so forth. That's what we're talking about. Macroevolution is the origin of species. Okay? We have a speciation event. And all of this stuff, we're talking about genetics and the gene pool, allele frequencies and genotype. Remember this from 120 P's and Q's and 2 alleles and 1 alleles and all that. That changes in these speciation events. Okay. Now, one of the very first lectures, I think it maybe was the first lecture, we talked about what a species is, about the biological species, morphological species, phylogenetic species concepts. And we said, basically, at the end of the day, what makes a different species is that it's a group of animals that reproductively isolated from another group. Okay, they're not sharing genes, so therefore their morphologies are different. Okay, so here are the type of things that keep species separate. Why do green frogs and bullfrogs not breed together? Well, these are some of the things that keep them separate. Okay, two general types. Then we have, what is a zygote? You guys remember what, or know what a zygote? What's a zygote? So that's a really early stage of development. Right after fertilization, we have the zygote. Okay. So these type of mechanisms keep species separate by preventing mating from ever happening. Okay. So if these two groups don't ever mate, they will remain reproductively isolated. And then we're going to talk about a couple of things. Even after two individuals mate, these mechanisms can keep them from being reproductively isolated, or keep them from being reproductively compatible. Okay, so pre-zygotic, post-zygotic. So probably the most obvious one here is if an example would be tigers and lions. Okay, we all know that these are two different species, right? Very obvious species, reproductively incompatible. But yet you can take the two and they will mate together and they can produce offspring. And in some cases they can produce fertile offspring. I can't remember if it's a liger or a tiger can be. But in nature, they never do breed together. Why is that? Where do you tend to find, what kind of habitats do lions use? Yeah, lions are more like savanna, open grasslands, these kind of things. What about tigers? They're a forest animal, right? So why do they not just eventually blend together? Because they have habitat. They are isolated by their habitat use. They don't come in contact with each other, okay? So they're separated by habitat. Their DNA tells them, okay, I'm going to use forest habitats, I'm going to use savanna habitat, therefore we remain separate, okay? So these are the things that keep dogs and cats. You'd be surprised, or maybe you wonder how often times people say, can a dog and a cat hybridize, right? What? I mean, yeah, you know that they don't. But why is it impossible for a cat and a dog to hybridize? We're not even talking about habitats. Obviously, they use the same habitat, which is living rooms, right? But what would happen biologically if a cat and a dog tried to breed?
Wouldn't work.
Why wouldn't it? You're right, it wouldn't work. And that's just
A
like,
cat could not.
don't be a dumbass, number one. But why doesn't it work?
Like, it wouldn't actually be fertile, like having a child, or like, not child, like having the offspring.
A puppy kitty?
Yeah, it wouldn't.
Cat?
It just,
Kitty?
like,
Puppy?
genes figure out, and it wouldn't become like fertile, and it wouldn't work.
If you look inside, and I don't know, you can decide who's the sperm is and who's the egg is, I don't know. Probably be a dog, dog and sperm. According to my dog, like anything that is not moving is fair game. But if you look in the cat's egg, right, haploid or diploid, one copy or two copies of chromosomes? Haploid, right? Haploid N equals 20. I don't know, we can look it up. my guess is they're gonna have a totally different diploid number of chromosomes. What happens upon fertilization? Those go from haploid to diploid, right? Every chromosome finds its mate, finds its partner, and then they use the genetic material to determine how to develop. So if your chromosome number is totally different, you're not going to develop at all, right? So that's another reproductive isolating mechanism. If you can't, the sperm and the egg don't combine and don't produce a fertile offspring, then you'll remain different species forever and ever, right? Out here, probably right in this, well, not in this pond, but there are ponds in Cherokee County where all these species of frogs occur. Why do they remain separate from each other? Because they don't breed at the same time, okay? This is temporal isolation. So wood frogs and green frogs would never be reproductively active at the same time of the year. So therefore, they're not going to hybridize and mix together. While it may be possible, their chromosomes may match up and they may be able to produce fertile offspring. They don't because they breed at different times. That's a temporal isolation. Okay. Here's another one, is behavioral isolation. Okay. I don't really have time. This pulls up.
The 0.2, 10, 12 pairs. They'll be on La Plata Island of the Ecuadorian coastline. La Plata Island, very small. So those boobies are dancing. Look at the booby dance. Look at that, the march of the booby. Taking its wings. Then they also sing, tails up. Then they do this.
Birds
It's all part of the
we see this. This is the male, the female's over here. She's trying to decide, is he worthy of her egg? Right? Think about the amount of energy, particularly the female, has at stake here. She wants to pick a good mate, right? She's about to be fertilized by the male, and then it's a lot of investment. So she wants to pick a good mate. So how does she first, probably the first thing that an animal needs to decide is it the same species, right? So one of the ways that birds pick and choose is based on their behavior of the potential mate, right? They do dance, they have song, and so that if that changes, the female won't recognize him as the same species, right? So those behavioral patterns are something that can isolate different groups. Okay. How do I know if you're in the right species? I'm going to watch your dance. Do you do like this with this foot? All right, that with this foot, so on and so forth. And those can act to isolate. Different species. Now, so breeding at different times, breeding in different habitats, using different mating behaviors, those will prevent mating from happening and therefore keep species separate from each other. Sometimes these things break down, right? You find yourself in the right habitat at the right time and mistakes are made. Two species will hybridize, right? But even after mating can take place, and even after fertilization sometimes happens, right? These hybrid offspring, if they don't develop and they don't become fertile, then they're the end of the line, so to speak, okay? These are those things. So the zygote may die, okay? That goes back to that chromosomal mismatch. And it won't grow into an adult. It could be that the hybrid byproduct is sterile, right? That's a good example of that one is this, right? Why are horses and donkeys considered different species if they can hybridize and they do regularly? Because the offspring can't breed, they're sterile, okay? So therefore, because think about it, these are obviously very different from each other, and the mule is a mix of the two. And if we just kept doing this, then you sort of lose the distinctiveness and it just blends, right, into something new. But that doesn't happen because of what we call hybrid sterility. And then at other times, it's basically mating happens, fertilization happens, development happens, and even the offspring are fertile, but they have very reduced fitness. So they're not really like either parent, they're a mix of the two, and they're just not adapted to anything in particular. So they just don't really have the opportunity to breed much at all, okay? And that will tend to keep species separated from each other, okay? All these things are talking about what keeps species distinctive. So what was in our example, what mechanism kept all of these different anole species separate from each other? On here, what do you think? What do you know kept all these different, what were they, Crystaltatus and Sargrii, and what are the mechanisms that kept anole separate?
Was it habitat?
Partly, I would think it's habitat, right? So if those big, huge canopy species are breeding on the canopy, and then one down in the grass, they're just not going to have the opportunity to hybridize. Oh, okay, so yeah, habitat. We didn't really talk about temporal. What about behavioral isolation? That's that dewlap, right? Do you ever see anoles? They do push-ups and they puff their dewlap up. So that is really important too. If these things change, then we could be talking about species splitting and separating from each other. Okay? If your legs get a little longer, right? If we go back to this and we look at this kind of change, right? This is the same species. We move them out there and their legs get shorter. Is it likely that that's going to make them a different species? Well, I don't know why that would affect any of those reproductive isolating mechanisms. Chances are if you took a nice healthy female from here and put them with a nice healthy male from there, what are they going to do? They're probably just going to breed, right? There's nothing that is keeping them separate, okay? Those are the kind of things that have to change in order for a species to split. So speciation then is either A, one species splits into two, so that would be, I guess you'd say, something like this. We have species blue, we have speciation back here, and we have the evolution. This is speciation right here. Or you could alternatively, maybe one species just as time goes on changes into the different species. Right? This one's a little more problematic because it's kind of like saying, well, yeah, that guy can't interbreed with them, but it doesn't have a time machine, right? Regardless, we're talking about the birth of a new species, okay? And it can happen in two different ways, right? Speciation can happen either allopatrically, right? Allopatric speciation is where the groups split when they're separated geographically. Allopatry. This means separate. we're gonna see this other one in a minute. Sympatric. Separate or spatially, geographically. You can think of means like simpatico or sympathetic, or symbiosis means together. So these two groups become isolated from each other, and then they, through time, become different species. Okay? And we'll get some examples of that. This is by far the most common way that speciation happens, is in an allopatric situation. Okay? Allopatric.
The evergreen and deciduous forests of California's coastal ranges and the western side of the Sierra Nevada form a habitat rich with amphibian life. But one species found in these forests has become very important in the world of evolutionary science. The photographs you are seeing are all Ensitini ischoltzi, the Ensitina salamander, from various places in the mountains surrounding the San Francisco and San Joaquin valleys. What makes this tiny 3-inch long amphibian so important to biological science? Each of these salamanders is able to freely interbreed with neighboring populations, except one pair. At the southern end of the valley, near the Tehachapi Mountains, the habitat of two subspecies, the yellow-blotched Ensitina and the Monterey Ensitina, overlaps. Yet we do not find any hybrids between them. They cannot interbreed. Why is this? The answer lies in evolution. The Ensitina salamander is an example of speciation in action, the gradual evolution of one parent species into two distinct daughter species. The ancestral population of California's Ensitina salamanders entered the coastal ranges from Oregon's Cascade Mountains. As the species spread further south, the Central Valley formed a barrier where the salamander could not inhabit, so the diffusing populations became isolated from one another, one moving into the Sierra Nevada and the other into the coastal ranges. As they continued to spread southward, each population faced unique environments to which they had to adapt. They were subjected to different criteria of natural selection. With the passage of time, and many generations later, the descendants of these two populations converged again at the south end of the valley. By that time, they had genetically drifted so far apart that interbreeding was no longer possible. In effect, they had become two separate species. This is perhaps the best known example of what is called a ring species. A parent population radiates around the central barrier, and by the time its descendants meet on the opposite side, they can no longer rightly be considered the same species as one another, were it not for their similarity to their common ancestor. This is at least one way that new species are born, and is an example of evolution in action. Ancestral population of California.
So this is a really cool example of speciation. Again, go back, this is going to be an essay question. I'm just going to tell you on the test. Okay, I'm going to ask you about this ring species and about why they split and why are they considered the same species. So I don't know if you guys are familiar at all with California. We've got the Central Valley. A lot of our food is produced down here. We have the coastal mountains, and we have the Sierra Nevadas. Okay. Salamanders like cool and moist. They don't like hot and dry, like in the Central Valley. So they come down, they migrated down from the north. We can tell from phylogenies because populations in the north are the basal ones. They split off first, and then the populations down here split off later. Right? And we see the color here, what does the color in this represent? Is just to say that if you look at the genetics of individuals in that area, they're all sort of related to each other. They're similar. You can think of this color as like genetic similarity. Okay, so here they come, there's migrating south, animals, populations migrate, right? And what happens, why did they split right here? Why did the split happen? Why the split?
Is that geographical barriers right there?
They don't, this is inhospitable to them, right? Here's good habitat, prime real estate. And guess what? There's no competition because nobody lives there. There's good habitat over here. So, but we see already, we go from pinkish, whatever, I don't know, what do you call it? Pinkish purple? The kids tell me I'm colorblind, but this is purple, these guys become pink. So already we start to see some changes in the genetics. Okay? Why do you suppose? And then again, this is where Tuesday's lecture comes in. Tuesday's lecture explains why we start to change from what are the microevolutionary mechanisms that cause changes in genetic makeup. One is natural selection, okay, which we've been talking about a little bit today. Why would natural selection cause differences in colors here? What do you suppose about the environment here and then the environment over there? You think it's going to be exactly the same in the Sierra Nevadas as it is in the Coast Range? So what could be different? What are some, in the anoles we talked about whether they have grass or big trees or bushes or whatever. But what are some things that may be different over here that are different from over there? What do you think, Summer? Shelby, what do you think? What could be different here? What could be something in the environment here that would make it different from the environment over there?
Different predators.
Totally, it could be totally different predators over here, different competitors, it could be different diseases. This might be limestone and this is granite, different pH, different food items. Anything that's different in the environment will lead to this process of an advantage, so therefore changes in genetics, right? And then there are other ones that you can hear about. But, so here they go, they start proceeding down the mountain range, right? And as they go, they change more and more, become more and more purple, right? Very different environments down here than it is up here even, okay? And by the time they get down to the bottom, very, very different from each other. Okay? Now, why don't these guys become pink? So why do genes that are over here not move into this populations? If we have pink genes over here, why does the pink stay over here and the blue stay over there? Okay, what would lead to blending was interbreeding, right? So what would make the pink move over here is it's individuals from here would need to breed with individuals from over there. So it's not so much that they couldn't breed, it's just if I pick them up and brought them over here, they might be able to breed. It's just that there is no movement of individuals across this inhospitable area. And that's called gene flow. There is no gene flow. Now, the genes can flow this way, all right? And they could flow all the way up to here, and these guys could breed with these guys and these guys and these guys, right? But they're not going to move across. So they're getting more and more different, more and more different. When they get to the bottom, these populations cannot interbreed with each other, okay? So in that way, it's like they're different species, but they are not actually different species, okay? Why not? Because they can all interbreed with each other. The only two that can't interbreed would be these two down here. It's a little confusing, okay? So again, they're all considered Ensatina eschaltzii because they are all connected around the ring by gene flow, right? Now, if you remove the segment here, and this became a totally separate population, and they kind of interbreed with those, these two would be different species. But because they're all connected with gene flow, they're still considered the same species, okay? But it's a really cool example about how splitting happens and how divergence takes place, right? And then it informs us about speciation. So once they become, and so what do you suppose would be different about this animal and this animal that would make them not recognize each other as different species? What kinds of things would have to change as they move south that would make them not be reproductively isolated or reproductively compatible? What kinds of things would have had to change? What, I'm sorry? Yes, any one of those. So these guys are salamanders. They're different from the lizards, but something like that, right? Something that, and in this case, it's probably like a chemical thing or maybe a visual thing. That so when the female looks at a male and says, "Is this the one?" Something that she uses to signal that he is of the same species has changed. So one of those reproductive isolating mechanisms has evolved. An example, it's not really speciation, but it's probably right on the way to being speciation. Two different species of darters in the genus Ethiostoma. They live, one species lives here, one species lives there. They're very closely related to each other, and they probably evolved when climate changed and they became isolated in these two different river systems. So think about an individual from here. It would have to swim all the way down into the Mississippi River and then up over here to get and have the opportunity to breed with another individual here, right? So they became geographically, allopatrically isolated and then evolved through natural selection and then became different species, okay? And we see rivers change a lot. Maybe you have individual species that lives inside of this river system and they're all freely interbreeding, but then what happens when you have an earthquake and a mountain arises here, and this becomes one watershed and this becomes another watershed, and then what we see is species W will split into W and E, okay? So that's allopatric speciation. Everybody's together, something happens, they become separate from each other, they diverge, and then they're reproductively incompatible. Okay? That's what has created all the species of anoles, all the species of every animal that we can see. Right? It's a little bit more difficult to see how sympatric speciation could happen. Okay? So one population develops into two or more when they're all together in the same geographic area. Okay? This is a little bit more difficult to conceptualize, right? There is some examples how this could work, right? And one of the most common ways that sympatric speciation happens is it's a huge mutation that causes a doubling in the number of chromosomes, okay? Called polyploidy. So where you basically get a chromosome number beyond the diploid number, okay? So maybe during meiosis, instead of the chromosomes separating like they should, you end up with an egg that has 4 times the number of chromosomes. And if you end up the same mistake over here, and then there's a sperm that has that, those mutant sperm and egg can be compatible, but everybody else is incompatible. Okay? So you result in an instantaneously self-fertile species that is reproductively isolated from either parent. This is a good example, something you want to read over in the textbook. But it's basically an instantaneous speciation. These two species look very similar to each other, but they have different calls. And if you look inside of the cells of Hyla versicolor, they have a tetraploid. So in other words, say they have 3 different chromosomes, chromosome 1 through 3, they'll have 1, 2, 3, 4 copies of that one, 4 copies of this one, and then 4 copies of the third chromosome. Hyla versicolor or Hyla chrysosceles has 2 copies of every chromosome. So obviously they're not compatible with each other, but they're self-compatible within each species. Okay, so this is just a mutational type of thing. You read about this thing called adaptive radiation, and it's not a separate idea, right? It's an example of allopatric speciation, but it's an issue of scale. This allopatric speciation at a big scale, very fast speciation. Many new species evolving quickly, and so you end up with lots of species being produced by this process. Okay, so islands are good. So actually the anoles that we've been talking about, there's a lot of species of anoles in the Caribbean because there's a lot of little islands. So there's a lot of opportunities for populations to become separated and to diverge from each other into new species. Okay? The anoles are a great example of an adaptive radiation. Okay? Because again, what better way to become allopatrically or geographically separated than the birth of a lot of islands? Okay. In Hawaii, there's this group of birds called the honey creepers, and they are very diverse, lots of different species. And they, if you look at the family tree of honey creepers, you'll see that they are different groups are related to these different islands. So an island is born, somebody invades the island, it then adapts to all these different habitats and speciates, and then another one colonizes a different habitat or different island, right? Diverges, just like the anoles happened. Okay. Here's a phylogeny of a group of salamanders, and let's look at how quickly these things are diverging from each other, right? Lots of different species, speciating very quickly. Again, here's the time scale. Very quick, very short branches, and so very rapid speciation. Here, the thing that is unique, here's islands, here's mountains, okay? These animals, kind of like the Ensatina, they only live on mountaintops, so if you have a bunch of mountains being born, then you have invasion of those mountains, separation of species, or populations, and lots of speciation happening. This is the sort of implications or the application of this. Speciation and new species are really important in terms of something like management, disease control. Lots of diseases are spread by mosquitoes. Right? Do you know that we used to have malaria here in the United States? Right? Used to be really kill a lot of people. How did we get rid of the malaria is caused by a protist that lives in the gut of the mosquito. So we got rid of the mosquito, okay? But you have to know what species of mosquito you're dealing with in order to control the mosquito, right? And so when the mosquitoes evolve and split into species, that's a really important piece of information in terms of controlling the disease, right? So this is just a small example of how this process is really relevant. So Tuesday's lecture talks about small scale changes. Today we moved into how those small changes accumulate and lead to the separation of species. We talked about what are the things that have to change. So if a species grows spots, one population evolves spots. Does that make it a new species? Say you have 2 ions. Well, we talked about the iron K and those experimental ions, right? You have the lizards out here, lizards here, so they grow shorter legs. Does that make them a different species? No. Why not?
It's the same species, just adapted to shorter limbs.
At the end of the day, the only important thing is if the male and females from these 2 places interbreed, they're the same species. Chances are having a little bit shorter legs is not going to make you reproductively incompatible with the original population. Now, what if this is a really shady area, and this is a really sunny area, and we transplant some anoles out here, so these guys have bright dewlaps. What's likely to happen to the color of the dewlap when we put them in a sunny environment? It's probably going to get darker, right? Now what happens then when you take the individual, now does that make them a different species? Could that make them a different species? Okay, so say we go, again, it's always like this. We have some with really dark colored dewlaps from here, some really bright colored dewlaps. If these are the ones that survive, right? And so as time goes on, we see a shift in the color of dewlaps. If we take these really bright colored offspring and bring them back here, is it possible that these guys are not going to recognize them as the same species? Possible, right? That's the kind of thing. If we see a change in the breeding season, if they start using totally different habitats, and then when you bring them back, they never come in contact with each other, those are the kind of things that have to change in order to result in a speciation. Okay? That generally happens. Why is it more likely that speciation would happen allopagically? Okay? Why is it more likely? Why does it make much more sense that if we have a population, population A, why is it not likely that population A is going to split into A and B right here on the same island? Why is it much more likely if we take A from here and put them out there, that A will split into B and A right here? That's allopatry. Why does it make more sense to have allopatric speciation than sympatric speciation? What are the things that cause this divergence when you put them out on the island?
They can't breed the originals.
Okay, so gene flow is basically, unless it's like, okay, it's breeding season, I'm gonna get in my little boat and just swim back over here and find a mate, right? So number one, gene flow is really limited because you're separate from each other. That's one thing. Why did we see the legs change when we moved them out to this island? Well, we basically, the scientists created that allopatric situation. Why did the leg length change?
There were bushes instead of tall trees.
Because the environment was different. Which leads to natural selection. The nature changes, right? What do you know about the environment over here? All species A, they're all in the same environment. Why would natural selection take place? Makes no sense, right? So, sympatric speciation is pretty difficult to happen. They're in the same place, gene flow can happen, they're in the same environment. Why would natural selection take place? Right? And then adaptive radiation is just it's underneath allopatric speciation because it's an example of allopatric speciation. It would be like this. Here's, I guess, the best way for me to explain adaptive radiation. Clearly, we can see how A can split into A and B if there's an island that gets colonized. But what if we have lots and lots of islands? Right? A now splits into B and C and D and E, so on and so forth. See what I mean? It's just that there's a lots of new environments to colonize, so therefore you see lots and lots of splitting at a pretty quick, rapid rate, right? And this happens a lot. It happens more than you think. Islands, mountains, streams, lakes, things like that. Here's another really cool example of adaptive radiation. In Africa, Lake Victoria, there's these fish in that lake that have radiated very quickly. Okay, so we have okay, here's the Lake Victoria. Here's the water level. Here's all the fish. Okay, trying to click. Not doing a good job. Here's all those fish. Are they sympatric or allopatric?
Sympatric.
Sympatric. Now what happens with climate change, right? Now suddenly, where we had high water level, the water level drops. And guess what? What have we created? A whole bunch of allopatric populations. And what happens? They diverge. This lake is humongous. I'm not talking 10 feet between here and there. We're talking 10 miles from here to there. And then you can create all these separate little incubating areas. A mutation pops up here. Oh, that's a good mutation. It becomes real common. This environment's very different. Divergence happens there. Then what happens? The rain falls again, water level goes up. What happens to all those populations? They come back in contact. But alas, I now breed in May, you breed in June. I now think that blue spots are sexy. You think that red spots are sexy. Anything like that. I use high in the water column. You now eat something that's at the bottom of the lake, so we don't have the same habitats. Boom, you've created 20 new species, right? That's speciation, and that's how it happens. And that's all I'm going to say about that. You guys are awesome. I appreciate you. If you have questions, just email me.